Hello students, today I am going to teach class 12th subject biology chapter 6. Chapter name is the molecular basis of inheritance. From this chapter our topic is the DNA fingerprint. What is DNA fingerprint? Printing is a hybridization technique used to identify the similarities of DNA molecule of two individuals. The technique of DNA fingerprinting was developed by Alec Jeffries in 1985. Okay. This technique is also called DNA colony hybridization by Guilfoyle in 1988 because in the technique is a known sequence of DNA in allowed to hybridize with a colony of DNA fragment extracted from a organism. DNA fingerprint, DNA profiling is generally used for the establishment of the identity of a criminal from blood stains, semen, etc. for establishing parentage in case of dispute and many other applications. The data from this approach are extremely reliable as compared to the conventional analysis of serum proteins and erythrocyte antigens and proteins. In this technique is the DNA isolated from any source will be subjected to southern blotting. The procedure of DNA fingerprinting. The procedure of DNA fingerprinting, what is procedure? The first of all, the DNA of the organism which has to be tested in isolated, it is host DNA. The DNA may be obtained from blood, semen, vaginal swabs and bone marrow. The amount of DNA needed for developing fingerprinting is very small. The isolated host DNA is then digested with a suitable restriction enzyme and the digest is subjected to the gel electrophoresis. These enzymes cut the host DNA into small fragments. The fragment of double-stranded DNA are denatured to produce single-stranded DNA by alkali treatment. The DNA fragments are separated by gel electrophoresis. Electrophorused DNA is transferred from gel into a nitrocellulose filter paper where it is fixed by baking the filter at 80 degrees Celsius for 2 hours. A known sequence of single-stranded DNA is prepared. It is the probe DNA. So it is the procedure of the DNA fingerprint. Then application of DNA fingerprint. Identification of criminals is forensic laboratories and legal pursuits. Settlement of disputed paternity. What is means of paternity? Who is the biological mother of the child? It is useful in restoring health of patients suffering from leukemia or blood cancer. It can explore the evolutionary pathway of a species. It can verify the kinship of an individuals with another. On the basis of DNA fingerprint, killer of Sri Rajiv Gandhi, the former Prime Minister of the India, was identified as the Dhanu, the sexual relationship of Bill Clinton and former President of USA with Monica Lewinsky. A working staff of White House was also concluded to be true on the basis of DNA fingerprint. The important terms and concepts. The main concept of the Chirgaff rule. What is Chirgaff rule? Rules in DNA molecules of every organism, the ratio of adenine, guanine, cytosine, and thymine is constant. Okay. Then, what is central dogma? Central dogma, the transfer of genetic information from DNA to messenger RNA, and again from messenger RNA to polypeptides. So it is called a central dogma. Central dogma. Helicases. What is helicases? The enzyme which unwinds the DNA helix. Denaturation, separation of two strands of DNA through heating. It is called denaturation. And in pharmosome or spleen. What is spleen? In eukaryotic, the messenger RNA come out from nucleus in the form of a RNA protein complex. It is known as spleen. Then last is the cistron. What is cistron? Functional unit of genes which produce a complete molecule of polypeptide. So it is in about DNA fingerprint is completed. Then I will upload in LMS its a topic related to questions. Okay, you can solve. Okay, thank you students.